Hi, everyone. Welcome. Welcome to the NFcast Bioinformatics podcast, where we discuss the latest advancements at the intersection of biology and informatics. In this podcast, we celebrate the brilliant people and organizations that are changing the world through advancements in genomics and life sciences. And our guest today is one of those brilliant people, I believe. His name is Cedric Notredame. Cedric has a degree in biochemistry and genetics at Toulouse University and did his PhD in bioinformatics at the European Bioinformatics Institute near Cambridge, of course. He became a senior principal investigator in the bioinformatics and genomics program at the Center of Genomic Regulation in Barcelona. We know it as CRG, of course. Cedric's notable scientific achievements to this day have been Nextflow, which is very near and dear to our hearts, and the T-coffee multiple sequence alignment algorithm. His work has received over 39,000 citations, which is more than I have. Cedric has co-authored a book written for me, I believe, Bioinformatics for Dummies, and it has sold over 40,000 copies worldwide. So Cedric, welcome. Glad to have you here today.
Thank you. Thank you so much, Rob, for inviting me and having me here. And I'm really looking forward to this.
Yeah, my pleasure. So you recently were presented with the Swiss Institute of Bioinformatics Resource Innovation Award at the Basel Computing Biology Conference for Nextflow. And you wrote a blog post at that time, Flowing in Love with Data Again and Again. It sounds a bit like a song. So we'll show a link to that blog below here. But the blog discussed the history and the evolution of the Nextflow project. And you were at the very beginning of the project. You were one of the main protagonists in the story. I'd love to start there at the beginning of the project. So tell us how Nextflow got started.
It's a long story, actually. It took about 6 years almost from the day we had the idea that something needed to be done along these lines till something really materialized. And interestingly, it all started with Anna Tramontano. And some of you listening this may have heard about Anna, who has been a major founding figure of bioinformatics. And Anna died, unfortunately, 2 years ago, too young. And Anna once told me, Cedric, I once mentioned to Anna, I'd like to do some computation on Amazon and all these things, exciting, all these cloud things. This was very new at the time. And then I said, I have just the guy you need. It's a master's student. He was a computer scientist. He decided to turn into a biologist. You should take him in your lab. And I don't remember Anna giving any bad advice. And this one was especially good because this advice was actually Paolo. And Paolo came in my lab. And he worked on this crazy project at the time, which was benchmarking the Amazon cloud with dollars. So he was running T-coffee and just measuring in dollar how much computation had been done. And this led to quite a nice paper called Cloud Coffee at the time. And so we stayed on the idea that we had to do something with the cloud because it looked like this was a nice way of powering up your computation when you needed it. Where every once in a while you need a lot of computation, poof, do it on the cloud, and the rest of the time you do it locally. Paolo was excited with this, and he started trying. But at the time, the Amazon cloud was really not designed for this kind of thing, especially the memory was not properly shared, and this was causing. So you had the choice between getting things that were running fast and crashing rapidly or running very, very slowly, but very stable. And that was not good. But at some point, we thought we had something. And we proposed Jamin, one of my PhD students, to run a massive computation he had to do. And this was, he was computing alignments and trees, and he had to compute millions of them. And so we prepared the fattest, strongest instances on Amazon and most expensive as well. And we launched the computation. And my money started burning, flames coming out of them. And nothing was computed. Everything was stuck because of some dependencies. I was horrified. Paolo felt humiliated, which is good because this is how he found the strength to do all these things. And Jamin was really pissed off and decided that he was not going. He only had 3 or 4 months left for his PhD, and he really had to finish stuff. And so Paolo, as any good scientist, turned his humiliation into something very, very positive. And that's really where Nextflow, that was a founding moment of Nextflow, where he knew he had to find a solution.
And all these cloud issues got resolved. I don't want our friends at Amazon hanging up the virtual Zoom phone at this point. Things worked out great in the cloud. But so tell us in your words, Cedric, what is Nextflow?
So Paulo hates it when I say this, but to me, Nextflow really is a kind of HTML for pipelines. Let me explain. HTML, like you have some text, and in order to make the title look like a title, you frame it with HTML tags, right? And this is going to say, well, this is a title. And you're not telling the browser how the title has to look like. The browser is going to know. Well, when Paulo started working on this pipeline language, the idea was that we were not going to rewrite all the pipelines. We had to use everything we had. We had to use every line of code we had because this is too much work and too much money to re-benchmark everything. And so the idea was that you had to be able to take an existing pipeline and to cut out the bits and pieces of computation, computing the alignment, turning the alignment into the tree. And you had to be able to put some kind of tag around these things telling this is receiving this kind of data and this is going to produce that kind of data. And that's why I call it HTML for pipelines, really. But the real smart idea Paulo had was that rather than using the usual flow, which would have looked like Makefile, really, he stumbled on this reactive programming thing. And that was really, his really smart idea on this thing to turn everything upside down, because this is very low level and this is very technical, but it makes everything amazingly scalable, because every unit of computation doesn't need to know what is going to happen or how it fits in the big picture. Every unit of computation simply needs to do some computation, and that really changes everything.
Right. Certainly the adoption of Nextflow in the community, which started in the lab and then spread to Europe, and then now we're seeing it across the globe. And the European was very much research institutions and academia at the time. But how did you get the first part of the process going, which was just engaging people in the lab to use this tool, get them to buy in? How did that work out?
So that's the thing, you don't get big if you don't start small. And to start small, you really have to engage your colleagues. And your colleagues in a lab, especially PhD students, they have no time to lose and they have no time to waste being instrumentalized as guinea pig for one project or another. So as a PhD supervisor, I was certainly not in position to tell the students, and now you're going to use Nextflow because we have to use Nextflow. No, that had to come naturally. And the reason it came naturally is because the idea, the proposition sounded so attractive from the start. You have this pipeline, and if you make a little bit of an effort to use the Nextflow syntax, then suddenly it can run as smoothly as you want on the computer, on your laptop, on the Amazon cloud if you want, or on the CRG cluster. And that was a naturally appealing prospect for the students. And that's really what made Nextflow successful from the start, because Nextflow was being used by the people for whom it had been designed. And I really credit entirely Paulo for achieving this level of engagement. And one of the things that worked, so Paulo loved making live demonstrations. And live demonstrations are usually a recipe for this. He still does. I'm sure I cannot imagine that he would have stopped. And live demonstrations are usually a recipe for disaster. Even the simplest thing, they stop working when you make them live. This is a law of the universe. I'm sure that if Schrödinger cat was making live presentation, then they will stay alive on each side or something like this. It really is difficult. And Paulo, for some reason, and that's probably not a random reason, always managed to do perfect live demonstration. And that really engaged people in thinking, wow, cool, if this can be done in 5 minutes like this, I can do it as well. And that's how it all started. None of the people who got really involved in Nextflow were supposed to work on Nextflow. This was not supposed to be their project. They jumped in because they found it so exciting. And that's especially Evan. That's the case with Evan Floden.
Right. Yeah, we're going to come to Evan in a little bit. So it sounds like that fact that it solved a problem, the fact that it was simple, really drove that adoption within the lab. And I think, as we talked before, that's what really started to drive the community engagement outside of the lab as well, right? The involvement by various European research and academic institutions, and that started to snowball from there. And I was around with in the HPC world, and you were our customer at Univa back in the day with Grid Engine, so we were kind of following what was going on, and we did some of the early work with containers with you folks as well. So you talked about cloud and Paulo's early experiments and maybe some of the lack of early success there. But, as containers came along and as the cloud came along and as Nextflow came along, all this kind of became possible, right? Can you talk a little bit about that?
Yeah, I mean, this was, so let's be clear. Paulo proved to be extremely forward-looking in the lab. And I still remember the day he told us Docker will be a thing. And at the time, Docker was nothing. Docker did not really exist. We had tried that stuff on Amazon, and it was difficult. And then Docker came along. But it's only, I think, 2 years later that Google said that Docker was a thing. And the same with code repositories. We had the chance to have Paulo, who really had an interest in how the field was moving. And while we were also lucky, we were also lucky that our expectations were met with the way the technology was developing. And things that were possible on paper, but not yet possible in practice, gradually became possible in practice. And that made a huge difference. But of course, and as you mentioned, nothing would have happened if Paulo had not also engaged the community. And this Nextflow mailing list has been amazingly active. And it's been Paulo single-handedly for many years, managing an entire community. And that was really, really something that worked great. But I don't know if we can say, when and how it became a high impact project. And that's really something that happened shortly before we published the paper, which is something you may want me to touch about a little bit. Or
yeah, well let's go there, because Evan Floden showed up, right? And I think things started to change in the project at that point. And tell me about how Paulo and Evan worked together, how that changed, and where the project went from there.
So as I was saying, so we had this great pipeline language, which was very useful in the lab, and we had to publish it, because you have to publish these things. It's my lab is an academic lab. If we don't publish well, it looks like we have done nothing. Now there were at the time. So when we started, we thought we were the only one, because SnakeMake was not published yet. So I'm sure it existed with its own community. But when Paulo really started Nextflow in 2012, I think was the first pilot version. I'd have to look. That was not known. The only alternative was Galaxy, and Galaxy was very complicated. And then there was CWL, and it was. Galaxy was not complicated, but Galaxy was not really designed for the kind of high throughput computation we wanted to make, and it was not really command line driven and all these kind of things. And so this was great, and we were going to have a very nice pipeline language, but we did not have something exceptional that could be given to the community as, look, we are solving problems nobody can solve. And that changed with Evan, because one day Evan figured out that Evan was very interested in all this virtualization. And he figured out and told me, we were having a pizza actually with Paulo and Evan on Parallel, which is a famous avenue in Barcelona. And so if you do something on parallelization, you want to have a pizza on Parallel, right? And so we were having a pizza. And Evan mentioned, there is something strange. When I run all these pipelines, all these RNA quantification pipelines on different flavors of Linux, I get different results. And I thought, wow, this is crazy. And I said, but you sure it's not random seed? No, he said, everything is controlled, same version, same random seed, everything should be exactly identical. and it's not perfectly identical. And I say, well, let's have a look. And he was right. You will be running exactly the same analysis on two different flavors of Linux, and you will get slightly different results. Why is that? Think of a computer as a machine with every line of code being a moving part. This is by far the most complex machine we've ever created. It cannot be stable. And these tiny fluctuations, they are neglectable from a scientific point of view. But if you are making a medical treatment, if you're making something that has to be robust, it has to be the same. And it is Evan who nailed it. And that really was a selling point for pushing the paper in Nature Biotech.
Oh, that's interesting. It's a big evolution now followed as well. But let's talk about some of the first customers that came on board. I think SciLife Labs in Sweden was one of the early customers. Many of you will know Phil Ewells, who's there and integral to that, and also integral to NF Core. Were you involved in those early customers and bringing them along? And how do you see NF Core fitting into the picture now?
Well, NF Core is amazing because now we are doing this. We're involved in a European project called BovReg, which is about improving farm animal breeding. I understand that eating meat is a problem these days, but it's not like this is going to stop overnight. So if we want this thing to be as sustainable as possible, we have to figure out ways to improve breeding, to fasten the improvement of genetic breeding. In this way you have animals that are going to be more efficient and can be treated better. There are a lot of things. And so NF Core is essential in the harmonization of data analysis that is required by these projects. We must have everybody running the same pipelines everywhere. And what NF Core brought to us was that we made a language and they turned the language into a standard. And if Nextflow was a drug, a molecule, I'd say that NF Core is the pill in which you put the molecule and you give it to as many people as possible. And so the increase of outreach that NF Core has given us is amazing. And it makes, a lot of things suddenly become possible. But it really is a construction for NF Core, for people as good as the SciLife Lab people to feel that they had to put their forces on Nextflow. Nextflow had to be very good. And NF Core is absolutely great, which is then making it easier. I think NF Core was one of the reasons to feel the energy to move into DLS2, which is making Nextflow modular and all these type of things. So all of these things really there is a strong synergetic relationship between all these projects. And then it's absolutely amazing to see these things becoming alive, and then out of our hands, really.
Yeah, we're definitely seeing a big shift in the marketplace. When I first started getting involved with Nextflow, people that were coming to us for the commercial offerings or for help with open source were Nextflow users, and now we're seeing people come to us because they see the beauty and the robustness and the richness of all of these NF Core pipelines, and they're saying, hey, I'm going to use Nextflow because I've found 51 pipelines on the NF Core website that look like some of them can help me and such, right? So we'll post a link to NF Core. I'm sure a lot of you know about those pipelines, but yeah, we're seeing that as a major driver for adoption now, and just really helping customers kind of leapfrog from, oh, I have to build this pipeline for RNA-seq or for eager, and no, I'm just going to take one off the shelf and get to work and start getting scientific results. It's a big difference.
And you'd think places like the EBI are skeptical, but they are not. They are very supportive. For instance, within the BovReg project, we had this discussion with Peter Harris, who takes care of pipelines at the EBI. But the question put to our little community was, should we go for the EBI pipeline or for the NF Core pipeline? And the answer of the EBI was very clear. The EBI pipeline are great, but they are great when data is totally stable and stabilized, entirely public. But all the intermediate layer, it has to be more agile, more flexible. And NF Core pipelines are great for this. You You can integrate the data as it comes. You can explore different ways of managing the data, of analyzing the data, and that's what you want. So this is, and it's interestingly, and of course, two things really. It's a set of golden pipelines that you want to use all the time, but for us, it is also a standard, and all the new pipelines we are doing, even if they are not necessarily meant to become part of the core of NF Core, we still make an effort to develop them at NF Core standards so that they can be interoperated with NF Core, and that makes a huge difference.
Yeah, we're seeing that as well. And there's just so many best practices that have come out of NF Core that people are now using, in our organization and in our customers' organizations. Where you're right, it's not necessarily a pipeline from NF Core, but it's following those best practices, those ways of doing things for testing and modularization and all those other kinds of good things. We're seeing a lot of that actually with the COVID effort as well, right? There's Nextflow pipelines for identifying variant strings and sequencing the virus for therapeutics and vaccine programs. It's very widespread. Some of that's with NF Core pipelines. Some of that's been developed. Well, a lot of it's been developed in the UK and elsewhere, of course. But tell us about some of what you're seeing there with the COVID efforts.
Yeah, so something amazing, and it's so I understand that about 50% of the COVID data has been going through Nextflow powered pipelines or NF Core powered pipelines, and it does not surprise me. And I don't think this happened because people love Nextflow or NF Core. This happened because these were the easiest tools to deploy. These were the easiest solutions for problems that were amazingly urgent to solve. And it's absolutely amazing how rapid this effort has been, how rapidly we managed to integrate as a collective endeavor, how rapidly we managed to integrate such massive amounts of data. And I'm not going to say that this was made possible by Nextflow because this will be an exaggeration, but I'm extremely proud to think that Nextflow, this project we developed for us, for the needs of a tiny lab, that this has provided some kind of contribution to this essential effort. And I still think that one of the reasons it has been so successful with the community is because of the way it started. It started with little money, and it had to be useful immediately. We discussed this before with the students, to engage the students. You were not going to engage the student with something very, very complicated that required 2 months of learning and training before you could run anything. You were going to engage the students if somebody reasonably learned in Linux could just take a template, plug in their own stuff, and get something working in a half day. That's how you engage people. And that's one of the reasons we engage such a large crowd, because these are effective solutions.
Yeah, I think it was all about getting immediate results or as fast as possible and reusing what was out there. We're still seeing that in the COVID effort. Just in the last couple of days, I've been on calls talking to people about how they want to push that variant identification out into almost the street level, out in the field labs and the public health labs and those kinds of things across the globe, so that they can do identification much, much faster than in central organizations like the CDC, et cetera, right? So we're seeing a real shift, and we're seeing more and more adoption of all these NF Core tools, but other pipelines as well that are going right out to the field level for use. So I'm going to shift gears a little bit. We're not done yet. I want to talk about, jump into the rapid fire section. So I'm going to ask you a question. You give me your thoughts in about 60 seconds or less, just really quickly off the cuff. So
that's a great opportunity to say something stupid.
Yeah, that's I'm setting you up basically. Exactly. So what will be dramatically different about the way bioinformatics will be performed in the next 5 or 10 years? What's going to change in bioinformatics, in other words?
I'm going to say something very lame, but it's obvious to everybody. This deep learning is really changing everything. Nobody expected that deep learning would work so fast and so well. And you can ask yourself what happened. Is it because there's no real breakthrough on the algorithmic side? The networks, the neural networks, they are not so different from what they used to be 20 years ago. But the computers have changed, and the data has changed. And I like if you think about AlphaFold, everybody thinks that AlphaFold has solved the protein folding problem. No, they have not. They have done something even better, because AlphaFold is taking on one side multiple alignments and on one side PDBs. And multiple alignments, what are they? They are experiments made by nature where you can see which mutations will preserve a fold. And PDB are experiments made by human to see a 3D structure. And AlphaFold is just liquefying all of these experiments so that they can be combined. And now, thanks to AlphaFold, we talk the language of evolution. So that's something extraordinary. And this is just starting. There is not a single aspect of biology that will not be impacted by deep learning. And how does Nextflow fit there? It's because all of this neural network, it's about processing data so that it can be fed to some neural network process. And the processing of the data is where action is happening now. You acquire data, you process data, and these things are having a huge impact. The most ambitious project I know of now is the Earth's biogenome. 1.5 million eukaryotic genomes that are going to be sequenced over the next 10 years. All of this data has to be processed and will be probably fed to some bigger than ever neural network. So this is something that is really changing bioinformatics and biology.
No, that's fascinating. We're seeing lots of machine learning and deep learning, certainly in the customer base right now, and real practical applications for precision medicine where they're applying machine learning models. So I'm going to shift gears and ask a little less serious question. Do you have insights as to why Paulo hates pineapple on pizza so much?
It's very clear.
He's so angry.
Yeah. It's very clear. Paolo, like many Italians, never recovered from the fact that Marco Polo brought pizza from Hawaii. It's very clear, on one of his trips from Hawaii, came with his pineapple pizza, and there it was. And I don't think the Italians ever recovered from Marco Polo traveling from Hawaii.
Yeah, I think Twitter's an outlet for him to vent on that topic.
That's the only rational explanation I have.
I like it. What are your favorite movies?
Any one of Wes Anderson, really. I'm a big fan. Life Aquatic being above everything.
Okay, I'm going to have to check that out because I'm not familiar. What are your 3 favorite go-to tools to get your work done at CRG?
Well, as we were saying, without Nextflow, my lab is dead. Without the Google tools, with Gmail and all these kind of things, I could not survive. And that's about it. And Zoom, obviously, who could survive with Zoom?
Especially these days.
I have no share and no interest. Disclaimer. But yes.
Cedric, this has been fun. It's been really interesting for me. I quite enjoyed it. We've talked about sort of the history and the evolution of Nextflow from the lab, from the early days. In the next episode, I'm going to have Evan Floden come on and maybe defend himself, but also talk about where sort of the next level. So Nextflow moves from being a community project, and it's still a community project, but also now there's the commercial entity, Seqera Labs. So we're going to talk a little bit about where things have gone from a commercial perspective and where Seqera Labs is going in the next episode. But I do want to thank you again, Cedric. Really enjoyed speaking with you, and we'll talk again soon.
Thank you, Rob. And let me say how proud I am to have seen Nextflow. You guys just raised this 2 Chan Zuckerberg Initiative grants for a project that started in a tiny lab. This is amazing. So thank you for inviting me and giving me a chance to talk with you about all these things.
Well, congratulations as well on the success, because you were instrumental in all of that as well. So thank you.
A little bit. Bye-bye.
See you. Thank you very much.
See you. Yep.